Hi everyone, today we're going to be solving AQA GCSE Biology Higher Tier Paper 1H. In this particular video, we are solving June 2020 paper. This question is about photosynthesis. Complete the word equation for photosynthesis. The word equation for photosynthesis is a pretty common question during the exam. Alright, so what will be the answer for this one? First of all, we will think of carbon dioxide, which is one of the reactant. Then carbon dioxide reacts with water. This produces glucose along with oxygen. So guys, this is a word equation which you need to memorize and keep in mind. Also, a good idea would be to memorize the chemical equation as well. Describe how energy for the photosynthesis reaction is gained by plants. The word photo literally means light. So obviously, we will get the energy for photosynthesis from sources of light. So light will be captured and absorbed by chlorophyll in the chloroplast. Students investigated the effect of temperature on the rate of photosynthesis. The students shone light from a lamp into conduit and measured the volume of oxygen produced per hour. Table 1 shows the result. We can see the temperature was increased from 20 degrees Celsius to 45 degrees Celsius, and the tests that were done in the rate of photosynthesis in cm cube per hour. We could see at 20 degrees Celsius the volume was 18.5, then it increased to 38.6 at 35 degrees Celsius. After that, at 40 degrees Celsius it decreased in a rate of photosynthesis, and at 45 degrees Celsius it's a very low rate of photosynthesis. The average is done for all of them except the first one, so the x needs to be calculated. To calculate the value of x, we will take these three values, we will add it up, and then we will divide it by 3. If there is a anomalous result that we can see which does not meet the trend, we can then eliminate it and also do the average. Draw a ring around the anomalous result in table 1. So to determine the anomalous result, it is best to go through that particular, within a table, to go through all of the lines, alright, for each test at each temperature. If we notice in this 45 degrees Celsius, we can see 14.2 is an unusually large value, whereas the other two results are 1.9 and 2.2. So 14.2 will be the anomalous result. Since we have circled so many things, let's eliminate all of them and label this one as anomalous result, easier to understand. However, in your exam, do not circle any other things other than the anomalous result. Suggest one possible cause for the anomalous result. Now, you see at 45 degrees Celsius, the enzymes are denatured, which is why the rate of photosynthesis is so low. Now, at 14.2 being this one of the value, means the temperature was not high enough to 45 degrees Celsius. We can easily conclude that. So, because the question says that we have to suggest a possible reason, so there can be many possible answers. One of the answer is the temperature was changed, or different mass of conduit was used, or the conduit did not acclimatize to that particular temperature, or we can say there was oxygen in the syringe before starting the experiment, or we can say the lamp or the light was moved, maybe it was moved a little bit closer, which caused it to have more photosynthesis within that same amount of time. Now all of these are possible correct answers. However, the answers that I like the most would be temperature change, had different mass of conduit, or conduit had not acclimatized. These are the better points, I believe. How did the students deal with the anomalous result? So obviously, the obvious answer for a anomalous result will be give one factor that the student should have kept constant in this investigation. Now, in this investigation, obviously, there can be other factors that can affect the rate of photosynthesis. They are light density, carbon dioxide concentration, conduit size or the amount of the conduit, and the conduit species can also affect the rate of photosynthesis. Any one of them is a possible answer. So guys, choose one of them and, stick with that particular answer for future reference. Table 1 is repeated below. Why did the rate of photosynthesis decreases from 35 degrees Celsius to 45 degrees Celsius? So we already talked about this particular point as 40 or 45 degrees Celsius as being too high. So since the temperature is too high, enzymes are denatured. So we're going to say the enzymes lose the shape of the active site. Complete figure 1 using data from table 1. You should label the y-axis, use a suitable scale for the y-axis, and plot the mean data or the average data from table 1 from 25 degree to 45 degrees Celsius. Then we are asked to draw a line of best fit. The temperature in degrees Celsius is already mentioned. Now we need to, on the y-axis, now y-axis, remember, is always, it is our result. So rate of photosynthesis in cm cube per hour. We will say mean rate of photosynthesis cm cube per hour. Now we will see the scale. The maximum value in our scale for mean rate of photosynthesis is 44. Means 45 is our maximum. Now we will see the units, how we can divide it to 45. So if we consider these 10 boxes as 10, then 20, 30, 40, then this will be 50. That's how we can get to our 45, which is going to be here. When doing this, we just need to make sure that we are using the graph in a way which suits us the best. Now, let's say because the lowest rate of photosynthesis is as low as 2.1, so having a value for 0 for this axis is important. As you can see, for the other axis, it is starting from 20. It's starting from 22.5, which is okay. So that can be considered as a graph break. Our graph starts from 25 degrees Celsius, and at 25 degrees Celsius, we have 33.2. At 30, we have 44. At 35, we have 40.8. At 40, we have 22. And at 45, we have 2.1. Now, the question says that we have to draw a line of best fit through this particular graph. So, generally, a smooth curve is the expected from the examiner. So, let's try that. Well, it is almost correct. The auto correction is not that great. Yeah, we can consider this to be okay. After this graph, we do not have any further things on this particular question. So we're moving to the question number 2. Diffusion is an important process in animals and plants. What is meant by the term diffusion? So we will just define what diffusion means, and the question has 2 marks. So diffusion is the movement of particles from an area of higher concentration to area of lower concentration. Figure 2 shows part of a leaf. So we can see the guard cells that are forming the stomata. We can see the air spaces, and we can see how carbon dioxide is diffusing into the mesophyll cell, specifically the spongy mesophyll cell. Molecules of carbon dioxide diffuse from the air into the mesophyll cells. Which 2 changes will increase the rate at which carbon dioxide diffuses into the mesophyll cells? Decrease number of chloroplasts in the cells. If the number of chloroplasts decreases, then this will decrease the rate of diffusion, because chloroplasts are the ones that are, absorbing the CO2. They require it for chemical reaction, for photosynthesis. Decreased surface area of the cells in contact with air. If we decrease the surface area, then diffusion generally decreases. It is a common idea. Increased carbon dioxide concentration in the air will increase the rate of diffusion, because there is a low concentration of CO2 inside the mesophyll cells. So CO2 will diffuse more faster inwards. Increased number of stomata that are open. If we open more and more stomata, then more CO2 will be available in those air spaces. So definitely it will increase the rate of diffusion for carbon dioxide. Increased oxygen concentration in the air. Well, oxygen will displace the carbon dioxide within the airspace within this particular airspace. All right. So definitely carbon dioxide concentration will become lower and rate of CO2 diffusion will decrease. So these 2 are the possible answers. Diffusion also happens in the human lungs. Figure 3 shows the human breathing system. We can see within a alveoli, the capillary, how it is carrying the blood in the capillary. Explain how the human lungs are adapted for efficient exchange of gases by diffusion. So in the question, what we need to focus is we are talking about adaptation, efficient gas exchange by diffusion. These are the key points that we are looking into for answering. When we are trying to answer, we need to think of the 6 marker, all right, as a level 3. So in level 3, we're going to get 5 to 6 marks. So how we're going to get to level 3? The relevant points with reasons are identified and given in detail, and they are logically linked to form a clear account of the answer. So at least the indicative point that I'm going to mention, you need to mention 6 points to get your full marks. So how we're going to go about the answer? First of all, we will say within the lungs, there are many alveoli which provides a large surface area. So this gives us 2 indicative points, all right? Many alveoli and large surface area is the reason for the many alveoli. Then we're going to talk about the capillaries are thin, which provides short diffusion path for oxygen and carbon dioxide. So oxygen to get into the capillary and carbon dioxide to leave the capillary. So this also gives 2 marking points. Then we're going to talk about the breathing mechanism that moves in and air into and out of the alveoli to bring in fresh oxygen and remove carbon dioxide, which maintains concentration gradient. So when we talk about it, we get several marking points in here, and then we can say there is a large capillary network around the alveoli. So the large capillary network has a good blood supply, which then removes the oxygenated blood quickly and brings carbon dioxide enriched blood more quickly into the lungs, and thus maintains a concentration gradient of CO2 going into the alveolar air and oxygen getting diffused into the capillary. Figure 4 shows a root hair cell. We can see the root hair cell with a very large protrusion. Alright, so this is the part which we call the hair, and it provides a large surface area. Water molecules are shown with dots and nitrate molecules are with crosses. Name the process by which water molecules enters the root hair cell. Movement of water through a partially permeable membrane is called osmosis. Nitrate ions need a different method of transport into the root hair cell. Explain how the nitrate ions in figure 4 are transported into the root hair cells. Use information from figure 4 in your answer. So first of all, name of the process. We can see there is a much higher concentration of nitrate ion inside the root hair cell compared to a lower concentration of nitrate ion outside. So active transport will be needed, and in the explanation we are going to write, because energy is needed to move nitrate ions from a lower concentration in the soil to a higher concentration in the root hair cell. Figure 5 shows 3 types of cell: bacterial cell 1 micrometer, liver cell 30 micrometer in size, and a mesophyll cell 0.05 millimeter in size. Give 2 similarities between the prokaryotic cells and eukaryotic cells in figure 5. So when giving similarities, we will see that they both have cell membrane and they both have cytoplasm. Other than that, we can say they all have ribosomes, and we can also say they all contain genetic materials which are made of DNA. So this will be the 4 points. Now you can think from here you can just choose 2. I would always go for cytoplasm and cell membrane because they are always common, and the next common thing that you can think of are genetic material, which is DNA, and ribosomes. Give 3 difference between the prokaryotic cell and the eukaryotic cells in figure 5. So we have to look for differences: prokaryotic cells and eukaryotic cells. So one of the thing we would say is that a eukaryotic cell consists of nucleus, a membrane bound nucleus where the bacterial cell doesn't have it, or the prokaryotic cell. By the way, the prokaryotic cell is the bacterial cell. Okay, so then we can think of other things like the eukaryotic cells have membrane bound organelles. The eukaryotic cells may have a large permanent vacuole. Alright, we can also say the eukaryotic cells have mitochondria where the prokaryotic cell doesn't have a mitochondria. The eukaryotic cells have chloroplast, the prokaryotic cells does not have chloroplast, and we can give this kind of differences and make the answer. Other points that we can also mention is prokaryotic cell is smaller. Now the question is asked in a way that give the difference between prokaryotic cell and eukaryotic cell, so we will refer to the answer in that way. Since the question says difference, so we will write only for a point for the first one, which is the prokaryotic cell. If the question would say compare, then we would write point for both of them. Now guys, we cannot use a pronoun for the prokaryotic cell in this case because using a pronoun may create confusion between the prokaryotic and the eukaryotic cell. And if a confusion is arise and an examiner finds that confusion present in your paper and that gives you a benefit of doubt, the examiner will never give you the number. So for that, we will never use pronoun, or we will try at least not to use pronoun where it is, going to create a obvious confusion. Calculate the ratio of the size of bacterial cell to the size of a mesophyll cell. Now, to calculate the comparison, we can see mesophyll cell is in millimeters, so we must convert it into micrometer. We'll multiply it by 1000, giving us a 50 micrometer size. Now, since we are asked to do a ratio of bacterial cell with mesophyll cell, so the mesophyll cell is 50 and the bacterial cell is 1, which gives us 1:50 ratio. Name the type of cell division that produce genetically identical body cells for growth and repair. So this process is known as mitosis, where the original genetic materials are preserved. Figure 6 shows a cell cycle. You can see it is divided in stage 1, stage 2, and stage 3. What percentage of time for one cell is represented by stage 2 and stage 3 together? So stage 2 and stage 3 consist of 3, 4, 5, 6, 7. Alright, whereas the whole thing is 20. So it will be 7 over 20 into 100%. So that should give us 35%. Describe what happens during each stage of the cell cycle. So stage 1, the chromosomes replicate and the mitochondria, ribosome or the subcellular structures increases in number, or we can say the subcellular structure replicates as well. In the stage 2, one set of chromosomes is pulled to each end of the cell, and in stage 3, the cytoplasm and the cell membrane divides to form two cells. So guys, as a general understanding, because the cell cycle data can be different for different, it can be very different in different textbooks, all right? And since the cell cycle is given in this way, we need to understand for our level, the stage 1 is representing DNA replication and the stage 2 is representing mitosis. Specifically, we are thinking of this phase of metaphase, if you are very specific about it. And the stage 3 is thinking of cytokinesis. If you want to do a little bit of further research on these particular points, you can do it definitely. However, because this particular data is like a lower class concept, so it can prove to be a little bit confusing for you. But don't worry, all right, it's manageable. You can go and do a little bit of further research. Lipases break down lipids, which two products are formed when lipids are broken down. Now we know that lipids consist of two things. It consists of fatty acids and it consists of glycerols. A lipid molecule consists of 3 fatty acids and 1 glycerol. A protein consists of amino acid, glucose is in carbohydrate, and glycogen is actually made by glucose. So that's why none of those are the correct answers. One model used to explain enzyme action is the lock and key theory. Figure 7 shows a model of that theory. We can see enzyme binding with the substrate, forming a enzyme substrate complex. We can call it the ES complex. And as a result, product is forming since the product no longer matches the enzyme's active site. They are leaving. Explain the lock and key theory of enzyme action. Use information from figure 7 in our answer. In the lock and key hypothesis, the understanding is enzymes bind to the substrate because they are complementary to one another. As you can see, the shapes are very complementary and fits like a puzzle. Once the substrate is broken down into products, the products are released because they no longer match the shape of the active site. There are many different types of lipase in the human body. Why does each different type of lipase act on only one specific type of lipid molecule? Enzymes are very specific, so we need to consider the answer in this way. Each active site has a specific shape, so it only fits one type of lipid molecule. This rest of the point is optional, so you may choose to write it or not to write it. The reason you may not choose to write it is because it's a repeat of the question, so it does not score any extra marks. Students investigated the presence of starch and glucose in the leaves of geranium plants. This is the method used. Place 2 identical geranium plants on a bench near a sunny window for 2 days. After 2 days, leave one plant near the window for 2 more days. Place one plant in a cupboard with no light for 2 more days. Then remove one leaf from each plant. Crush each leaf to extract the liquid from the cells. Test the liquid from each leaf for glucose and for starch. The question says, describe how the student would find out if the liquid from the leaf contained glucose. To test for glucose, we need to know that glucose is a reducing sugar, and any reducing sugar can be tested with a Benedict's test. So the student adds Benedict's solution to the liquid, and then the student will boil that particular test tube. All right, the student will heat that particular solution. If glucose is present, the blue color will change to brick red. Describe how the student would find out if the liquid from the leaf contains starch. To test for starch, the student will add iodine solution to the liquid, and if starch is present, the color will change from brown to blue-black. Table 2 shows the student's result. With glucose, leaf from the plant kept in light for 4 days, there was a strong positive result, and leaf from leaf from plant kept in light for 2 days and then no light for 2 days has a weak positive result. For starch, there was a strong positive result for the plant kept in light and negative result for the plant kept in 2 days for dark. Explain why the leaf in the light for 4 days contained both glucose and starch. Sorry, it's 4 days. Okay. Now, obviously, this particular plant were able to do photosynthesis, and glucose is made from photosynthesis. And since the plant produced excess glucose in photosynthesis, it converted those glucose into starch. Explain why the leaf in a cupboard with no light for 2 days did contain glucose but did not contain starch. So the plant that was with no light for 2 days, it converted its stored starches into glucose. So the glucose can be used for respiration and other metabolic processes. Because there is no light to make more new glucose by photosynthesis. Suggest one way the students could develop the investigation to find out more about glucose and starch production in plants. The student can repeat this investigation to test for glucose and starch in other parts of the plant, for example, the root or the stems, or the student can test the same experiment in other species of plants. The student can also measure the concentration of glucose and starch. The student can also repeat this experiment for variegated leaves. All of these are possible answers. I'm going to mention few points and you can choose one of the best point and repeat it for all of your answers for a similar question like this. Guys, for those of you who do not remember what is variegated leaves are leaves that has different colors in them. Since we know that only the green part of the plant produces glucose in photosynthesis and the other parts do not photosynthesize. So testing the same experiment for a variegated plant will give the student idea that only green part of the plant are able to do photosynthesis. Many plants have evolved defense mechanism. Figure 8 shows part of a gorse plant and part of a deadly nightshade plant. So in the gorse plant, we can see that it contains thorns, and in the case of a deadly nightshade plant, we can see that it has brightly colored berries, meaning that it is a toxic plant. The gorse plant has evolved to have sharp thorns. What type of defense response are thorns? Thorns are a form of physical defense mechanism because the thorns will poke the herbivorous animal and prevent them from eating the gorse plant. How do thorns defend the gorse plants? It prevents animals from damaging the gorse plant, or we can write it deters the herbivores from eating the gorse plant because it hurts them. The deadly nightshade plant has poisonous berries. What type of defense response are poisonous berries? Because the poisonous berries are a form of poison, so we can definitely say they are chemical barrier. They are chemical form of defense. Scientists noticed that in one area the gorse plant and yellow leaves had yellow leaves and stunted growth. One reason for yellow leaves and stunted growth is deficiency of nitrate ions in the soil. Explain 2 other possible reasons for yellow leaves and stunted growth. Do not refer to nitrate ion in your answer. So the first reason that we can provide is lack of magnesium ions. Since magnesium ion is needed for making chlorophyll, so our explanation will be not enough chlorophyll is produced for efficient photosynthesis, and as a result not enough glucose is produced, which is then needed to make proteins for growth. For our reason 2, we can say that there is an infection by pathogen, so the leaf becomes yellow, and as a result less photosynthesis, and as a result not enough glucose is present for making proteins for growth. Some added points will be, so first of all, the reason will be the plant got infected by aphids which remove sugars from the phloem, and again the answer will then lead to so not enough glucose to make proteins for growth. An alternative answer for reason number 4 will be there is a lack of available light or sunlight, so chlorophyll breaks down, and again the answer will be so not enough glucose to make proteins for growth. The gorse plant has nodules on its roots. The nodules are part of the living root tissue. Bacteria which convert nitrogen gas into soluble nitrate ions live in the nodules tissue. Figure 9 shows the nodules in the root. We can see the root nodules in figure 9. Suggest how the nodules benefit the bacteria. The bacteria are able to obtain glucose from the plant, and then the bacteria can use the glucose for respiration and making other substances like amino acids, which then can be converted to protein. Explain how the nodules benefit the gorse plant. The gorse plant are able to obtain nitrate ion from those root nodules, and nitrate ions are very important for making amino acids, which then can be converted into protein. For many years, drugs have been extracted from plants, which plant material was chewed as painkiller. We know that willow bark tree, where they produce a chemical which is similar to aspirin. Rose petals or foxglove plant or blackcurrant, they produce other chemicals, but not the pain-relieving chemical aspirin. Data from Million Women survey in the UK was collected for over 15 years. Scientists analyzed the data to study the effect of consuming alcohol on liver disease. The scientists include 400,000 women who regularly consumed alcohol, included 400,000 women who did not consume alcohol, and excluded women who already had a liver disease. Age and gender were two factors controlled in this analysis. Many other factors were also controlled, such as two other factors which the scientists would have controlled. Scientists may have controlled the BMI or the obesity level of the patient, their smoking habits, diets, and medication. The scientists may also have considered family history of liver disease, their fitness level, their ethnicity, and the area of the UK they live in. The data was analyzed for women who drank alcohol with meals, for women who drank alcohol not with meals, for women who did not drink alcohol at all. During the survey, approximately 1,500 women developed a liver disease called cirrhosis of the liver. Scientists calculated the relative risk of developing cirrhosis of the liver for each group who consumed alcohol. A relative risk of 1.0 means there was no such statistical difference between the groups who did consume alcohol and the group who did not consume alcohol. Figure 10 shows a summary of the results. So, alcohol consumed in grams per week and relative risk of developing cirrhosis of liver. So, we can see with people, those that had consumed 50 grams per week did not have a relative difference of 1. So, we can see a relative difference of 1 after they have consumed more than 150 grams per week. We can see there is a relative difference of 1. Now, let's see the question. A woman drinks 150 grams of alcohol per week, not with meals. The woman decides to change the drinking 150 grams of alcohol per week with meals. Calculate the percentage decrease in relative risk of developing cirrhosis of the liver for these women. So, if the particular person consumes without the meals the 150 gram, then the relative risk is 2.55. And if she chooses to drink it with meals, then the relative risk is 1.6. So to calculate, we will do 2.55 minus 1.6 divided by 2.55 into 100%. This will give us the percentage decrease. So 0.95 divided by 2.55 into 100% gives us 37%. Now this 37% is closest to the whole number, 37% decrease. One glass of wine contains 12 grams of alcohol. A different woman drinks 2 glasses of wine each day with her meals. Calculate the relative risk of developing cirrhosis of the liver for these women. 12 grams of alcohol consumed per day times 2 meals per day times 7 days per week gives us a total of 168 grams of alcohol consumed per week. With 168 grams of alcohol, so this is 168. Now we will extrapolate and touch the line with meals, and we find out the answer to be 1.8. So the relative risk is 1.8. Now obviously the extrapolation is important because the extrapolation will give us our mark. Figure 10 is repeated again, and consuming alcohol with meals instead of not with meals decreases the relative risk of developing cirrhosis of the liver. Give 2 other conclusion about the relative risk of developing cirrhosis of the liver related to alcohol consumption. So we can see if a particular person in general increases their alcohol consumption, they have a higher relative risk of developing liver cirrhosis. We need to do a data code to use because we have to use data from figure 10 according to the question. So we can start with, for example, with 50 grams of alcohol per week with meals is relative risk of 1, and with 265 grams of alcohol consumed with food has a relative risk of 2.5. The other data that we can say is that if we look into the extrapolation of this particular data, then we can see consuming less than 50 grams of alcohol per week with or without the meals does not increase the relative risk of liver cirrhosis. One optional point is even a small amounts of alcohol at 25 grams per week may increase the relative risk if it is not consumed with meals. We can see that particular data from here. By doing an extrapolation, the answer comes to 1.1 with 25 grams without meals. Suggest 2 reasons why the data is considered to be valid. The data is considered to be valid mainly because there is a large number of people used in the survey, and it is a very long term, like a 15-year survey, and it is well controlled with many controlled variables. So any two of these 3 points will be the answer. You can choose which ones you want to write. Suggest one aspect of the survey which might reduce its validity. People often underestimate their consumption of alcohol when they are asked upfront, or people may change their lifestyle habits over a period of time, or some people may drink all their weekly alcohol at once whenever they feel like. So this particular data then applied to those same people will be invalid. Cirrhosis of the liver leads to liver failure. Describe the effects of liver failure on the human body. This particular question can have a level 2 response which can score us between 3 to 4 marks. So when aiming for 4 marks, the answer should be scientifically relevant fact, and the events or processes are identified and given in detail to form an accurate account of the liver failure. So an indicative answer I will give you, all right, and you can choose how you want to go about writing your own answer, and you can just take some points from here, but please refer to all of the key points that I mentioned. So first of the key point will be no bile will be made in the liver, and we will then write its consequences. Fats or lipids will not be emulsified, and the surface area of fat as a result will not be increased. The pH of the small intestine will not be alkaline because bile makes the pH of the small intestine alkaline. The enzymes in the small intestine will not work efficiently or effectively, so food will not be digested. The person may lose weight. This particular response can bring about all of the 4 marks. However, I'm going to give you some other alternative responses as well. Now, an alternative answer for the one, with the destruction of the liver due to liver failure, we can give in this way. Lactic acid will not be broken down in the liver, and lactic acid will build up in the body. Since lactic acid is toxic, the body will be poisoned, and the oxygen debt will prolong in the body, which will lead to muscle pain. Guys, this sequence of answer will also give you 4 marks. So if you are choosing the first sequence, stick with that particular answer, but if you are choosing the second sequence, then stick with the second sequence of that answer. So guys, there are other alternative answers. I'm going to add in a slide and give you the answers in those slides. So another alternative answer for this will be, as a result of the liver failure, amino acids will not be broken down in the liver. So amino acids are not deaminated, and amino acids are not made into urea or ammonia. However, if any ammonia is formed, it will be toxic, which will not be converted into urea because the liver is damaged. So accumulation of the amino acid in the body will occur, and it will slowly further create metabolic problems in all of the cells. Guys, the next 2 points will be given together because alone they cannot score the 4 marks. Since the liver does not break down toxins like alcohol, toxins will accumulate in the body and the body will be poisoned. So jaundice or swollen liver can result from that. Since glycogen stores will not be formed in the liver, the body cannot control the blood glucose concentration. So as a result, hyperglycemia or hypoglycemia may occur. Guys, you can choose any one of these series of answers as your original answer and score your 4 marks. All of them are relevant. However, do not opt to write all of them in the exam. Only write one of the segment. Alright, so if you are going for example, if you're going for liver does not remove toxin, then write the consequences of liver not removing toxin and leading to jaundice. And because this by itself cannot score maybe the 4 marks, so you may also write the glycogen stores will not be formed, and then go for the consequences that body cannot control blood glucose and hyperglycemia or coma may occur. Monoclonal antibodies or MABs are usually made using mouse lymphocytes. Candida albicans infection produce serious symptoms in patients with a poor immune system. Recently, scientists have produced MABs to Candida albicans using human lymphocytes produced naturally after an infection. Candida albicans lives in the throat of the infected patients. A sample is taken from the throat of the patient with a suspected Candida albicans infection. The sample is transferred into a microscope slide. Describe how the MABs and a fluorescent dye could be used to see any Candida albicans pathogen on the slide. At first, the fluorescent dye should be bound with the MABs, and then we need to put that bound fluorescent MABs on the slide and rinse off. MABs will bind to the Candida albicans and show up in the microscope. Guys, for those of you who are wondering why the MABs will bind to the Candida albicans, because you see the Candida albicans protein are actually, the antigens of the Candida albicans are complementary to the MABs, the monoclonal antibodies that are produced from the Candida albicans. That's why they will go and bind with it. So if the particular sample contains, its sample taken from the patient contains a Candida albicans, then it will bind with it and we will see it under the microscope. Otherwise, we will not see it. Because it is a fluorescent dye, so we need to use UV lamp. In a laboratory, the human lymphocyte MABs were injected into animals infected with Candida albicans. The MABs caused increased phagocytosis of the Candida albicans pathogen. Doctors intend to start a trial to give the MABs to patients severely ill with Candida albicans. Explain how increased phagocytosis of the Candida albicans pathogen will help the patient. So, more Candida albicans or the pathogens will be engulfed by the white blood cells and killed by the white blood cells. Therefore, there will be less damage to the cells or the tissue, in that particular region where it infects. It has been shown that this MABs treatment is effective in the laboratory using both infected tissue culture cells and infected live animals. Then MABs treatment for Candida albicans is now ready for clinical trials on people. Describe how clinical trials should be carried out. Generally, whenever we are asked about a question of clinical trial, the answer needs to be scientifically relevant on the events or the processes are identified and given in detail to form an accurate account to score the maximum of 6 marks. Since our aim is to get 6 marks, so I'll give you an answer which wraps itself around for that particular 6 mark, and you need to understand that this question about clinical trial does not matter for any particular disease. It is similar for every diseases, for every medicines. So you just need to, you can do something, you can memorize it, all right, and use it for every answer that talks about clinical trials, all right, after it is used in infected tissue culture and infected live animals. If it is talking about that, you can answer the same answer for every one of them for any medicines and it will still be correct. So to carry out that clinical trial, it should be, first, the MABs treatment should be given to healthy volunteers at a very low dose to test whether it is safe and to test for its toxicity and side effects. Then the MABs treatment will be given to some patients with the disease. Basically, these are volunteers. All right, these are patients, those who have volunteered to test for correct dose, to test for any side effects, the efficacy of that particular, MABs treatment. And then it will be used in a double-blind trial where neither the patients nor the doctor knows who has been given the MAB and who has been given placebo, as to find out whether that particular MABs treatment is effective. Scientists have also used human lymphocytes to make MABs to other pathogens and to some types of cancer cells. Suggest one reason why these new MABs have been more successful in treating diseases in humans than MABs made using the mice. Since the body will not reject this new kind of MABs, the mouse MABs are more likely to be rejected as foreign protein, or we can say the human lymphocytes have already responded to that infection, so they are known to work against the disease. Any one of these 3 alternative answers can be written, so you can choose which one you may want to write. However, my personal favorite is the body will not reject the humanized MABs, the human MABs. Guys, that's the end of this particular question paper. Thank you for watching this video. Guys, best of luck for your exam. And guys, see you in the next video. Bye-bye.